Hello and welcome to the Treesearch and WWSC Science Center virtual conference series, a series that was initiated to contribute to the scientific discussion in a time when conferences have been cancelled due to the COVID-19 pandemic. The full program is found at the Treesearch webpage, where you also can watch the previous presentations. My name is Josephine Illegård, and I'm here to present the 8th speaker in the series. It's Torbjörn Pettersson from KTH, who will present AFM Beyond Imaging towards molecular understanding of cellulose interaction. Torbjörn is associate professor at KTH, where he focuses on studying the surface interaction and surface modifications. You in the audience can interact and ask questions or comment via the YouTube chat function. For that, you need to be logged into YouTube. Any questions will be answered after the presentation. That will last approximately 30 minutes. And with that, I hand over to Torbjörn.
Thank you, Josephine. I'm going to start sharing my screen, I hope. Okay. Thank you all and welcome to this presentation. My name is Torbjörn Pettersson. I'm associated professor at KTH and working a lot with AFM and surface interaction. Today's lecture or presentation will be dealing about understanding cellulose interaction using AFM. So the title is AFM Beyond Imaging Towards Molecular Understanding of Cellulose Interaction. A lot of my work and the work in our research group is dealing with creating new materials from wood, going from tree to nanocellulose. We're working with wood pulp fibers, and we're working with nanocellulose, CNF. And from CNF, we normally can make thin films or paper sheets. We can use the CNF in composites. We're using CNF for cancer diagnostic. We make foams. So we can use CNF for a lot of things. And we're now also working a lot on pulp fiber as well. But why do we want to understand the interaction between cellulose and cellulose? It has to do with when you're working with cellulose, it's containing a lot of water. When we want to put this into a product, we need to get rid of this water and remove it, and need to combine the cellulose fiber or nanocellulose with other type of material. And in this image, we can see two freeze-dried or two semi-images of a paper sheet. One paper that are freeze-dried in the middle, and to the right of that one, we see an air-dried paper sheet. And we can see a quite big difference when they freeze-dried or air-dried. So something happened during the drying. And during the drying, it's form a joint. And we want to understand what's actually happened during this joint formation. What component can give increased joint strength? What is the adhesion mechanism in the joint? What happened during drying? What happened if we put the other component in? What happened when the water evaporates? In the wet state, the fiber wall is highly swollen and an open porous network, and drying into a more solid material. To do this, we're using AFM, atomic force microscopy. Most of you probably know about AFM as an imaging tool. And in imaging, we use the AFM as a sharp tip, rastering over a surface, capturing data from different positions of the surface, building up a three-dimensional surface structure of the material so we can see the topography or some other type of interaction parameters during the imaging. And this three-dimensional structure is normally published as an image, or we can get a surface roughness parameter. But the AFM can do a lot of more things. We can also use the AFM to measure interaction, to measure the force between objects. And to do this, measure the force, instead of rastering the surface under the cantilever, the tip, we move the tip up and down and monitoring the deflection of the cantilever, as illustrated in this movie. We move the sample up and down, and we measure the deflection of the cantilever when it's bending and hooking to the surface and will be withdrawn. And capturing this, we get the data of deflection versus motion of the surface. And by knowing different parameters like spring constant, deflection sensitivity, or the hard wall contact or a point of contact, we can transform this data set into a force versus separation data set. And by looking at this force versus separation data set, we can tell more about what actually happened in a system, or what type of interaction are present in the system. To get better understanding of different material, we normally put a spherical particle onto the cantilever. We glue it with two-component glue or heat melting glue. And this type of measurement is called colloidal probe AFM, and was introduced by William Ducker in 1991 and have been used since then. And we use it a lot. We can put different type of particle. We can put cellulose particle, we can put silica particle, we can put polymeric particle like latex particle, polystyrene. We can put gold, we can put zirconia, we can put different type of metals, ceramic materials. And if we have like silica, we can also absorb polymers to it. So we can have a wide variety of different materials and measure. We can also put cylinders, we can put fibers, filaments. We have been using it for cotton fiber, hair fibers, etc. So one can measure a lot of different things. When measure, we can also get the information about the adhesion between the interacting materials. And adhesion can be due to many different things. The most fundamental is the thermodynamical work of adhesion. That's when we just have an interface, and when we separate the interface, we create two new surfaces. And the work needed to separate them is the work we gain when we delete the surface and create the two new surfaces. It's require work to separate the surfaces. We can also have mechanical interlocking. We can have different type of chemical interaction, hydrogen bond or covalent bonds. We can have some electrostatic interaction, or we can get sort of mechanical interlocking by interdiffusing one molecule into the other surface or vice versa. And by measuring the surface force profile, like the top left graph, I can see if I can get a laser pointer. I can move my mouse instead. Oops. Up here, we have a really snap off. It's basically a work of adhesion. If we look on the right here, we have a more jagged pattern. It's more interdiffusing or mechanical interlocking type of behavior. We can also measure the interaction by if we modify surfaces with layer by layer technique. And we get different forces when we separate, depending on the number of layers we have absorbed onto a surface. And it could also be an odd even effect depending on which materials are in the outermost surface. 2 years ago, we also used the AFM to try to illustrate what type of interaction we got in a cross-linked fibril foams. And by doing a measurement at different pH, we could indicate that the interaction was a hemacetal formation. I will come back to that type of measurement later on in this presentation. Here is an example. I have put in this presentation, I have put a couple of examples where we have used AFM and measure interaction to gain more understanding of what's really happened in the system where we have cellulose as one of the components. Here is an example where we wanted to work to find a suitable compatibilizer to use cellulose in a polymer composite. So basically get the polymer material to get better interaction to the cellulose surface by adding a compatibilizer. And we had a PhD student, Simon Utzell, who worked on polymerizing polystyrene polydimer block copolymer in micelle form, and this micelle form could adsorb to a surface, and by heat treatment we can get the micelle to spread out on the surface. And we wanted to measure how much interaction is it between polystyrene and a compatibilized surface of cellulose. In this case, we actually use silica as a model surface instead of the cellulose, but it works similar at the cellulose. So we adsorb the micelles onto the surface. We heated it up and cooled it down and went to the AFM and measure AFM interaction between polystyrene particle and the surface. And if we have a compatibilized surface, we had slightly higher interaction compared to the non-modified surface, but it was not that high increase in adhesion that we were thinking of when we did it in room temperature. Then we realized when we're working with composite material like polystyrene, we normally had some heating step when we mix it. So then we did measurement at higher temperature. And at high temperature, we basically didn't see any difference in interaction. We saw some difference with the compatibilized surface. We saw an increase with contact time. And this is most likely due to interpenetration of the polymer from the surface into the polymer matrix. It was an interesting result, but we didn't get what we really wanted to see. So instead, we did think again, what do we really do when we make a composite material? We actually blend the material at higher temperature, then we cool it down, and it's at the lower temperature that were interesting to have the higher strength. So what we did was to take the surfaces modified and unmodified, took polystyrene particles, spread them on the surface, heat them up, and cool them down. And then we went in with the AFM, find the particle, we put glue on the UV curing glue on the cantilever, went over the particle, went into contact, cured the glue, and pulled the particle off the surface. And when we did this, we get a quite drastic increase of the pull-off force from the compatibilized surface, like 700 nanonewton, and the non-compatibilized surface was 250 nanonewton. So it was a quite drastic increase. And when we now had the pulled off particle sitting on the AFM cantilever, we could actually look on them with SEM to see where we had the contact zone, where the particle had been sitting. We also could go in with an AFM sharp tip and do imaging and measure the surface topography where the particle had been sitting. And if we look on the blue images on the top, the unmodified surface, we don't see so much difference. A straight line profile, the red line, indicate there is basically nothing happen on the surface. On the right side, where we had the modified surface, we actually see a dip in the height where we had the particle sitting. We basically pulled off the compatibilized material from the surface. So if we want to increase this strength even further, we need to work more on the compatibilizer anchoring onto the surface. We have also been using the AFM to measure flexibility of never dried pulp. When you're pumping pulp fiber in the process, it's interesting to know how much energy it's needed to pump the fibers. And to get an understanding, it's good to know the flexibility of the pulp. And to measure on never dried pulp is a challenge. We need to mount the fiber over an edge and be able to go in with the AFM and press onto the fiber and see how much the fiber deflect and the cantilever deflect due to the force applied. And we need to get the fiber sitting on the surface. And if we were thinking different scenarios to get this fiber to sit on the surface. The easiest way would be to put the water-based adhesive, but water-based adhesive are quite nasty and affect the surrounding water quite a lot. So we were believing that the water or the glue will affect the whole fiber, not only the joint where we put the fiber on to a surface. So we ended up to use a clamp. So we had an underlying magnet, and put a steel slab on top of it to clamp the fiber over the edge. And the problematic thing is with the AFM, we actually have a roof over the cantilever, and the height between this is in the order of 100 micrometer. So we needed to have a really, really thin steel slab. We ended up with a 20 micrometer thick steel slab that we clamped the fiber with. And we were able to measure flexibility of different types of pulp, bleached softwood and TMP, for example. And they have different flexibilities, and we could also calculate some type of elastic modulus of this pulp fiber in never dried state. To continue, it's also interesting to know elasticity. We have used the AFM to measure elasticity on not cellulose to the left. We have used it on where it's an A, living fibroblast cells that were healthy. We also used cancer-induced healthy cells, or no, not anymore healthy, they were cancer-induced. And we used the same cell type that were induced with cancer with a drug. And we could measure on the cytoplasma and see different elasticity. And the biologists that we were collaborating with could draw different conclusions about the mechanism within the cell based on the elasticity measurement we did. Most of the measurement on living cells have been done on the nucleus, but we wanted to do it on the cytoplasm to see the actin and vimentin network within the cytoplasm. We have also used the same type of measurement we used for the cell on the cellulose model beads. It's cellulose dissolved in a solvent and regenerated in an antisolvent to make gel beads. And those gel beads have been used as, or are now used as model substrate to understand swelling and deswelling of cellulose and cellulose interactions. And we wanted to measure the elasticity of this type of gel beads if we used cellulose originated with different charges, with different charge densities. We dissolved cellulose with different charges and regenerated beads. It was Pernilla Karlsson who did most of this work, and I was involved in doing the AFM measurement and doing indentation in liquid state of this bead. And to speed up the process, we actually used the cantilever in air, and we have the bead in a liquid menisc. So we measured the cantilever in air, indenting into the liquid film and indenting the cellulose bead, and measure the pull-off force. And during the pull-off and indentation curve, we could fit to theoretical contact mechanic models and calculate and elasticity. And the elasticity is slightly decreasing when we increase the surface or the charge density of the cellulose used. The same type of elasticity, since we in the previous slide where we measured the flexibility could get some elasticity from the bending of this pulp fiber, we also wanted to do indentation into the pulp fiber in never dried state. So we measure elasticity by indentation of different type of pulp and could do it on never dried pulp. We have also used this type of indentation to measure on carbon fiber. This is not cellulose, but it's an illustration of what one can do by combining AFM with fine mechanical electron microscopy and focused ion beam. So we can do changing of the substrate. Here we took a carbon fiber, adhere it to a substrate and milled it with ion beam to get a longitudinal cross section like the top left image here. We also used the focused electron beam with Omniprobe to make smaller cross section where we measure on the transverse direction of the fiber. And we did AFM indentation, and we also sent the sample to Australia to do Nano-indenter and measure with Nano-indenter. the same material. And we had 3 different type of carbon fiber, and we measured the elasticity. And really good was that the AFM indentation were really good agreement with the Nano-indenter on the longitudinal cross section. When we looked on the transverse cross section, we have a really big difference in the measurement from 106 gigapascal to 54 gigapascal. And we started to think, why do we have this difference? Basically, when we do the AFM indentation, we have a non-hysteresis because we don't press so hard. With the Nano-indenter, they need to get the good data, they need to press much harder. And on the transverse cross section, there is a hysteresis in the measurement. In the AFM, we pressed much lower force and could get data from that. And we didn't have any hysteresis in the curve. So by having higher load, we get some deformation in the material. And it could be 2 different origins. Either we have the stacked graphene layer in the carbon fiber are buckling and shearing down, or we're moving larger flakes down. So we have an deformation, internal deformation in the fiber, as illustrated in the bottom right graph here. Additional with the indentation, we wanted to go a step further, a more challenging measurement, taking the gel beads and looking what happened when the gel bead is drying. And go in with the AFM and do similar measurement as we did in the 2 slides before. We had regenerated the cellulose bead, then we had solvent exchange the cellulose bead into ethanol or water. In the images, in the image series in the middle, the top row is water swollen bead, and in the bottom row it's ethanol swollen bead. They are original from the same type of bead, the same regenerated bead that have been solvent exchanged for a week. And then we went down and did indentation during the drying. The bead is decreasing in size over the time, and we needed to follow the bead when it's shrinking. If we look on the time scale for the ethanol swollen bead in the bottom, we can see in the third image it's only 4.5 minutes. So it's shrinking really fast. So it was really challenging to come in contact with the particle and indent, but we succeeded. And we could measure the elastic modulus during the drying at different drying time for the water swollen bead and for the ethanol swollen bead. And to understand this type of measurement, we have also involved SAXS measurement and WAX measurement, where we have studied the cellulose bead during drying in SAXS. And during the drying, from water, we can see 3 different phases from the SAXS curve. And we have interpreted this as the change during the internal structure changes in the cellulose bead during the drying. In the ethanol case, it's only 2 stages during the drying, and the internal ending structure is completely different. This work is submitted for publication, and hopefully it will come out soon. One of the last example I will talk more in detail about is where we wanted to evaluate the chemical bonds cross-linked in aerogels of dialdehyd cellulose or dialdehyd CNF. If we have CNF and dry it, it's normally collapsed. If we freeze dry it, we can get a foam, like the foam we see here in the C image. However, if we do some sort of cross-linking, we can get the water to evaporate here without freeze drying. But how can we do this cross-linking? There are different methods to do it. There have been work on BTCA and so on. But in this case, in this study, we were focusing on cellulose that we oxidized with periodate. And with the periodate, we formed dialdehyd on some of the glucose ring in the cellulose. And when we have an aldehyde and an alcohol group, they can form hemacetal. And this reaction is going to the right with the acid and is hindered in higher pH. If we have an hemacetal, it can react further with alcohol in real acid condition to form acetal. And indicative, we took the dialdehyd cellulose and freezed it and throw it in ethanol and dry it. And we formed foam-like structure. And if we put this foam-like structure in higher pH, it dissolves. But if we take this foam and put it more in acid alcohol, we actually can form from the hemacetal into acetal. And when we have done this step and put it in high pH, it do not dissolve or degenerate. If you have this hemacetal or acetal foams at pH lower than 12, they are water stable. So we wanted to understand, is it hemacetal that are formed or acetals? Can we do this with the AFM? And how can we do it with the AFM? And as you can guess, we did manage to do it. With the AFM experiment, we're set up that we, on a flat surface and then the colloidal probe, this silica sphere on the cantilever, we absorbed the CNF, both oxidized and non-oxidized, on different probes. And then we went into the AFM in liquid state and measure in different pH at more or less neutral, 6.5, and at high pH, and then back to 6.5. And then we added acid ethanol. And in the end, we went back to pH 6.5. So in the different stages here at pH 6, we hope to see hemacetal. At pH 12, we think we get back to this stage. And go back to 6.5, we're back on the hemacetal. If we add acid alcohol, we hope we're going into acetals. And if we have formed acetal and go back to pH 6, we will not break the acetal, and the formed acetal will be stayed in acetal stage. Here, we actually can see force curves measure on in the pH 6.5 to the left. For non-oxidized, we have a force versus separation profile that's a quite low force. In the oxidized case, we have a quite strong adhesive force, and the adhesion force is increasing with the contact time. And by looking on the integral of this curve, the force versus separation, we can get the work needed to separate the force, and this work could be called adhesion energy. And if we plot the adhesion energy to the right for the different type in contact of the different color of the lines, if we have where we think we have formed hemacetal, we can see a quite high force. So we actually need to break the CO bond in the chemical bond, the covalent bond. If we increase the pH, we get a decrease in this adhesion energy. If we go back to 6.5 again, we get it back to form hemacetal. So then the case is, what happened if we put acid ethanol? We have the surfaces separated, put acid alcohol in, we form hemacetal with alcohol and aldehyde on the cellulose surfaces when they are separated. And when we put them together back and try to pull them apart, we don't have an interaction because the acetal formed in the cellulose are blocking the possibilities of form hemacetals. And when we go back to pH 6.5, we still get the same type of interaction. The aldehydes are blocked with acetals with the ethanol from the solution. And if we look on the energy needed to break a covalent bond, a CO bond, it's around 530 kilojoule per mole. It's actually, if we calculate per bond, it's 0.88 attojoule. And if we look on the energy levels that we actually have, we have around 35 femtojoule. We can actually start to calculate how many bonds do we actually have in this contact zone. And by doing a few assumptions of how large the contact area is and so on, how much materials we have, how much degree of oxidation we have, we can calculate how many bonds per accessible dialdehyd glucose ring are we breaking in the different stage. For the oxidized case, we actually seems to break 1.4 CO bond in this type of back of the envelope calculation. And the same would be if we do the same calculation. If we assume it's hydrogen bond, it should be 12 hydrogen bonds that will break. So it seems to break something more than hydrogen bond. So it's highly believable that we actually break CO bond in the hemacetal. And it could generate up to 2 CO bond per glucose ring that's are oxidized. And we got a measure of 1.4, which is quite good. If we look on the non-oxidized case, we can't form any CO bond, and if we did the calculation, it should be 0.24, and it could be up to 6 hydrogen bond. It's slightly too much, but it might be some other type of interaction involving as well, not only hydrogen bond and aldehydes or hemacetal formation. But it was a quite interesting measurement where we can illustrate different type of chemical bonding in the interaction. And as a concluding remarks, I want to stress that AFM is a versatile tool, not only for imaging. We can do force measurement and understanding of interaction. Different adhesion mechanisms can be evaluated. Flexibility and elasticity can be measured in dry state, during drying, in wet state, or in never dried material. We can also monitor changes in material properties over time, like during drying. We get good agreement between AFM and Nano-indenter. The effect of additive or surface modification can be evaluated with AFM. Chemical surrounding can change the interaction, like in the hemacetal or acetal case. And for the hemacetal or the aldehyde cellulose, freeze-induced cross-linking indicate hemacetal formation and acetal formation with acid treatment. And most of this work have been done in collaboration with a lot of different peoples. And I want to acknowledge funding source like Biomac Innovation, Energimyndigheten, Wallenberg Wood Science Center, Biomime, and past and present member of the Fibre Technology Group. And special thanks to Professor Lars Wågberg, Per Larsson, Simon Utzell, Johannes Helvig, Hailong Lee, Pernilla Karlsson, and Johan Erlandsson. And thank you all for attending and listening. So I hand over to Josephine if there are any questions.
Okay, I got some technical problems. Torbjörn, are you Are you there?
I'm here.
Thank you for a really nice presentation. Really versatile tool, AFM. Let's see now if I get all the technical things. We open up for questions from the audience. So please type in your questions using the live chat. So far, we have no questions, but we will give the audience some time to write.
Yes.
Say that, yeah, Anastasia, it's a very nice presentation, and thank you very much, Torbjörn.
Thank you.
Speaking about Anastasia, I should also mention that the AFM is, you can apply for access and support for AFM measurements and the Treesearch research infrastructure support. And now we have a question from Jakob. Thank you, Torbjörn, for a very interesting presentation. I have a question on the beads drying. The ethanol cellulose bead E modulus decreases and increased with drying time. What may be the reason?
We have a swollen network that are condensing during drying and forming more condensed network. That's why the elasticity is increasing. I don't know exactly how to explain it more. We're still looking on. We have submitted a paper and we will see what the reviewer says about it.